Welcome to the Wheats on Your Mind podcast. I'm Aaron Harries. Wheats on Your Mind is brought to you by the Kansas Wheat Commission and Kansas Association of Wheat Growers. On this episode of the podcast, our guest is Allan Fritz, the K-State wheat breeder in Manhattan, Kansas. Allan has been the wheat breeder for how many years now, Allan?
I started on April 1, 2000, so almost 25 years to the day.
Okay, well, I would say you've got the program rolling really well, and I wanted to have you here in the podcast to talk about some of the focuses of the breeding program, some of the things that you're working on that maybe people don't know about that I think are really neat. But I think it'd be a good place to start and do a quick overview of the breeding program for people who might not be familiar with the size of a wheat breeding program. So K-State has essentially two wheat breeding programs, the one in Manhattan headed by you and one in Hayes from Guorong Zhang, obviously collaboration between the two. But tell us about the size of the program. You know, how many crosses do you make a year? How many people are on your team?
Yeah, so we make somewhere around 750 to 1,000 crosses a year within the mainline breeding program. There might be some other things that we do on the side that are side projects that add to that. Actually narrowed it down a little bit. We used to be about 1,200, but that gets to be almost too much to manage at times, and so we've kind of trimmed that back to 750 to 1,000. What that translates to is, roughly, 1.5 million plants in the field that we look at as individual plants. Obviously, we're not spending a lot of time on each individual plant, but selecting things based on disease resistance, plant type. And then as we move further into the breeding program, then we get into the yield testing. And so, we run somewhere probably in the neighborhood of about 25,000 yield plots per year. A fairly sizable program, but I think it's what it takes in order to have the numbers that give us the chance to have the success that we want to have in terms of successful varieties.
Yeah, you face a lot of the same stresses that a wheat farmer does. You've got to get those plots in the ground. You want to harvest as many of them as you possibly can, but at the same time, I'm sure you've had years where you've lost a lot of research just because of the weather, right? You've had to abandon some plots just due to drought.
The last few years have been pretty miserable, to be honest. We've had a lot of locations that we've abandoned. We've had a few more that we probably should have abandoned, but I need seed or data from somewhere. Yeah, so we normally are planting at 12 locations a year. I kind of depend on losing a few just from the standpoint of there's no way to harvest everything that we plant, just logistically, and we just know something's going to happen somewhere. But the last few years have been a little rougher with stand establishment and drought issues, and so we've been down to 4 or 5 locations in some years that we've harvested, which is not quite as many as we would like to have.
Got to take a good group of people to manage a program like this. How many people are on your team in different levels of all the way from student to professional scientists?
Yeah, so there's 4 people that are the staff on the project. 3 of them are full-time. The other is a 30-hour person, and they really do the nuts and bolts of everything in the program. And then there's also grad students who are here for their education and their training, but they are also labor at peak times of the year, and they also need to know how to run a breeding program, so having that hands-on experience. So we've run usually around 3 graduate students at a time, and then during the summer we'll have 3 to 5 undergrad students that are working on the project, usually only 1 or 2 during the semester, during the school year, where we're a little less intense in terms of the busyness, but a fairly large crew that it takes to keep everything going.
And equipment.
And equipment is not cheap either. So the small plot combines, planters, drills, sprayers, all of those sort of things. And so I'm really blessed with a crew that is exceptional at getting all that work done and maintaining the equipment and keeping everything rolling.
And the team has had success. I was looking at a chart that you shared with me that over the years, the genetic gain that we've seen with wheat. Tell us how far we've come in the last decade or two.
Yeah, so we started looking at everything that's been released from the Manhattan program since Carl 92. And so we started growing all that out starting in 2017. So now we have a pretty good database of yield, and we can show we weren't really making a ton of progress from 1992 to about 2009. And I'd love to say that I came in 2000. It takes about 10 to 11 years to release varieties, and so really that was just me. It helped as we got from 2009 to 2015. We're on a much steeper trajectory. That's really not true. Raleigh Sears was an awesome breeder. I think a lot of that has had to do with the change in the way we've run the breeding program, and the focus has gone from selection under ambient conditions to selection under fungicide conditions. And so I think that's allowed us to identify the things that actually have higher yield potential. 25 years ago, there wasn't a lot of fungicide use, but now there is. And so that's allowed us to take advantage of that. I think also using some of the CIMMYT germplasm has given us a bump in gain, but I really think a lot of it has been really that focus on just looking at yield and not confounding it with all the disease factors and using disease and observation nurseries to get that disease data and not relying on that out of the yield plot.
And CIMMYT, briefly tell us, is the Norman Borlaug Institution in Mexico, correct?
Yeah, so it's the International Center for Maize and Wheat Improvement in Mexico, and Borlaug was largely responsible for the Green Revolution, and so they've been a repository of genetics and advancement, and so we use their material for quite a bit of work on durable resistance to rusts, but I think we're also catching increases in yield out of that as well. So KS Providence has had a really nice yield record, and it's about 37.5% CIMMYT spring wheat by pedigree. So I think just bringing in some of that new genetics has helped us in addition to the disease resistance side of it.
Well, you mentioned the cycle it takes to create a new wheat variety, 10 to 12 years, doing conventional, traditional breeding methods. But you do some things that kind of speed that process up, try to get genetics through the pipeline faster. So explain to us kind of the difference between the normal breeding process and terms like rapid cycling and tools like double haploids, how those work and how those accelerate the process.
Genetic gain in breeding is a function of the time of the breeding cycle. And the breeding cycle is the time from when I would make a cross until we return progeny from that cross back to the crossing block. In our conventional standard winter wheat breeding, it's 11 years cross to release, but the breeding cycle is about 7 years in that case. After we get really the first really good year of yield testing, we might enter that back into the crossing program. So that's a 7-year breeding cycle. We can do things like rapid cycling, which is just using a method we call single seed descent, where we take a single seed from a segregating population and advance it, and we can do that 3 times in a year in the greenhouse. And so we can shortchange 2 of the years that we're normally spending in the field, just allowing it to get to the point where it's more true breeding. And so we can cut a couple of years off by doing rapid cycling. Doubled haploids are also another way of doing that. So really, when you make a cross and you grow that seed out, if you grow all the progeny from that, you're going to get all this range of types. You're going to get segregation for height and disease and maturity. After you do 3 or 4 generations of selfing, then the progeny begin to look like the plant that they came from. And so we've got to get through that stage to get to a higher level of homozygosity. And that's what we've been doing in a conventional program, just through self-pollination and just that wheat is naturally self-pollinated. So we just let it do what it's going to do, but we do that more or less at 1 cycle per year, 1 generation per year. In DHs, we can take that F1, it gets pollinated with corn. So I will say we finally found a use for corn. And the corn chromosomes get lost, and you end up with 1 set of wheat chromosomes. There's a chemical called colchicine that's used to double the chromosome number back, but now there's no segregation. Everything is completely uniform. So we've circumvented the need for all that self-pollination, and that will cut another year off. So we can actually get down to about 4 years for a breeding cycle instead of 7 using DHs. The challenge is DHs are provided by Heartland Plant Innovation. They do an awesome job, but they're also not cheap, so we can't run the same size of program.
Yeah, so getting back to that true breeding line again, basically you're trying to get all those plants to look alike, right? Because when you plant them, you're selecting for the additional traits. So traditionally that takes a long time of planting, selecting, replanting, selecting, and the DH just shortens that up.
It just shortens it up. Now we're looking at very fixed lines, and now it's like, okay, that's good enough to look at again, or it's not.
And when you reference putting something back in the crossing block after 7 years, that just essentially goes back and becomes another parent?
As a parent, yeah, because in breeding, we're just making progress, incremental progress over time, and building on finding something better, putting it back into the crossing program, and trying to find something even better than that. So it's a continuous process of improvement.
So you've also done some modeling, right? I mean, there's some predictive abilities to a breeding program now, and you talked about the goal is trying to reduce the number of crosses you make, reduce the number of plants, knowing already what's in those plants. So how do you do that? What's the process that you go around to do some modeling on this?
You can implement modeling in a lot of different ways, and a lot of programs are doing this, and we've done it as well. You can, more or less what you have is you have the phenotype that you collect in the field. That's the observed characteristics. So resistance or, particular low volume, that's a phenotype. And then you have a set of DNA markers that's run across the whole genome on all those materials. Then you can take a set of materials that you don't have any phenotype on, but you have the genotype data, and you can use that model based on the genotype and phenotype that you do have to predict those things you've never tested. Mm-hmm. So we do that. We've done that quite a bit with things like quality. One of the challenges, I think, is we really want to be able to double genetic gain, and implementing genomic selection in that way is great. It gives us some efficiencies in a lot of ways, but it doesn't really radically change genetic gain that much because we really aren't affecting the cycle time necessarily. So we've been thinking about some strategies. Really, I have to give credit to Jeff Morris, who was our sorghum geneticist here at K-State, is now at Colorado State. It's his term, genomic-assisted recurrent selection, but there are some things that we can do where if our model is really accurate, we can actually make that selective process 3 times a year. So in theory, we could go 21 times faster than we do now. Realistically, we're not going to be that efficient. There are some genetic things with that. You can't get your recombination too far ahead of the generation that you're in, so we need to be able to evaluate more often. So we're not going to have that kind of efficiency, but we can maybe we don't need to be 21 times better. If we were 3 times better, that would be awesome. And so we're really looking at some of those sort of strategies. I think the question is whether that's what you do for the mainline breeding program, or this is maybe how you rapidly change for a particular trait. If you really want to generate something that's outside of the norm, whether that's for drought tolerance or quality or disease resistance, I think that it's still an open question of how we use that, but we've seen enough evidence to this point to think that it's a good strategy for at least being able to rapidly move a trait into the breeding program and change the allele frequency in the breeding program.
Okay, so GARS,
genomics-assisted recurrent selection.
Okay, and you're targeting through that process gaining protein while not having a yield drag, right?
Yeah, that's one of the projects. So we've built several GARS populations. One is just adapted to central Kansas. One's adapted to western Kansas. One is focused on protein. So we've moved some genetics from wild relatives of wheat. We've evaluated those. We've selected things that have higher protein but still tend to hold on to their yield pretty well. We will have some yield drag, but we've selected those things, and now we're at the point where those are in the field, and we're really understanding how this is going to work and putting the wheels underneath it in terms of making the selections and seeing how effective this is going to be. But for the protein, obviously we have protein and gluten plants in Kansas that are looking at taking a bunch of bushels. And so it might be beneficial to really have some higher protein, but we need to drive that change rapidly. So we're hoping that that's what we can do with this.
Speaking of protein, we've heard for many years now the BX70E, which is kind of a super strong protein. You've collaborated with Oklahoma State on doing some work using that super strong protein. Tell us what that's about.
Yeah, so it's actually been around for a long time. It's the foundation of the Canadian super strong class. BX70E was found in a variety called Red River 68, among some other things. And so that was a spring wheat adapted to the Red River Valley of North Dakota and released in 1968. So it's been around. It's an overexpressed high molecular weight glutenin, and the glutenins are our storage proteins. What that does is it can give you extra strong protein. Now, BX7 alone doesn't do that, but it's sort of required in order to get that super strong trait. And so Snowmass is another variety out of Colorado State. It really came out of a breeding population out of the Hayes program that carried the BX7 gene. So that's another one that's had that. Brett Carver at Oklahoma State's worked quite a lot using Snowmass and built that into a Gallagher background. And these things just have extra strong protein. Just to give an example, normally we would say we would want a farinograph stability in hard wheat of maybe 10, 11 minutes would be good.
Farinograph is a quality test that's done on dough, and stability you're shooting for 9 minutes.
Yeah, 10 minutes would be good. So the two characteristics, so it's sort of the gold standard for quality testing. It's the language that the milling and baking industry uses. So the two things they're most worried about are absorption. So how much water does it absorb?
Because it's a cheap ingredient.
Because it's the cheapest ingredient, and it also affects product quality. Mm-hmm. And then stability. So how stable is that dough to over mixing? So like if you think something goes wrong in a bakery and you've got dough in a mixer, but you've got everything kind of backed up, how long can it sit in that mixer and over mix before you end up with a tub of goo, basically? But really that stability is a critically important trait. In most hard wheats, we'd be happy around 10 minutes. Some of these things have stabilities of 60, 70 minutes. And so they're well beyond what would be normal, and they would not actually work well in a bakery on their own. They would be way too strong, but they become high volume ingredients that you can use to improve doughs, let you take vital wheat gluten out of the mix, perhaps also reduce additives, which is a big deal as we move into an era where we're looking at getting rid of a lot of those additives. Can we find ways to maintain the product quality without? And this is a mechanism to do that.
Let's talk a little bit on another level of food health, and you've done a lot of work on grain fibers. You know, there's different types of fibers, beta-glucan being one of them, arabinoxylin's a different type of fibers. Tell us about the work you've been doing on both of those.
Yeah, so one is just pure serendipity. We were evaluating materials. Farinograph said that absorption is a critical trait. We'd be happy at 60% water absorption. Had a line come back from the quality lab with 67% water absorption. And I'm like, okay, something's wrong. You guys need to run this again. So Youngway's lab ran it again. And it's like, comes back 67% farinograph absorption. I'm like, well, I guess you didn't do anything wrong. And we started looking at that over a number of years, and it was stable over a number of years. And so as the Grain Fiber Coalition kind of came into existence, there was a question about, well, are there any unique genetics in the U.S. that might contribute to this? I'm like, well, look at this one. And so Devin Rose at UNL did the initial evaluation and identified that line as having about a 50% increase in arabinoxylan. And it also appears to be genetically different from the other sources of high arabinoxylan that are being used. So that line is actually on increase for potential release in 2025. Trying to figure out if there's enough fiber there to make that an IP trait, or if that absorption characteristic is sufficient enough for it to be an IP trait. But we're interested in that because we think we can continue to build fiber. The other one is the high beta-glucan out of barley. So beta-glucan is what gives oats and barley their heart healthy label. The Wheat Genetics Resource Center at K-State has actually taken the gene from barley and moved it into the wheat genome on all three of wheat's genomes. We're currently at the point of having all three of those stacked so that we can see what beta-glucan level we've gotten. We saw initial results where if we had just on one genome of wheat, we got a fairly minor increase and just an incremental gain if we had it on two of the three genomes. So now we want to see if we can have it on all three. Does that actually give us a bigger bump? And then can we add this to also the arabinoxylan and get to high fiber characteristics that would have some unique health benefits?
Just a little sidebar there. So wheat has three separate genomes, A, B, and D, and when you do your work, you can do it on just one or all of them.
It's one of the reasons why working on wheat has been such a challenge, because it's a hybrid between three separate grass species. And so there are three different grass species that contribute all of their genetics to common wheat, but there's similarity between each of those genomes. And so it makes it a challenge to work on, because if there's a gene for photoperiod response, for instance, on chromosome 1, it's going to be on the A genome, the B genome, and the D genome. You're going to have genes that are affecting that. So it complicates the wheat inheritance, which sometimes works for us and sometimes works against us.
It's a complex critter. So
it is.
Back on the beta glucan, people might recognize that as the heart healthy fiber that's in oats. So
right.
You said it's from barley. How do you get it from barley into wheat?
So that's the magic of what people at the Wheat Genetics Resource Center can do, and around the world people have done. You can cross wheat and barley. It's not easy, but you can cross them, and you can get a chromosomal translocation. So you can get exchange of genetic material between wheat and barley. There were addition lines that were available, and then the Wheat Genetics Resource Center took that and pared the segment down that's responsible for high beta glucan to a smaller segment that we've moved into wheat on all 3 genomes now. So it's through the magic of some special recombination genes and the skills of some people who are really good at crossing things that normally don't like to cross.
They're kind of the genetic whisperers.
Yeah.
They have a knack for it. Durham program. A lot of people might know K-State or not know that K-State has a winter durum breeding program. Durum being obviously the pasta wheat, and you've got several years in this now and actually have had release of a variety. So bring us up to date on your durum program.
Yeah, so Andy Ald is our durum breeder. He's been running the program for, I actually don't remember how long. It's probably been about a dozen years now that he's been in that role. He released a line in 2023 that we had been having under evaluation, and we have had our first commercial durum production in 2024. There was about 300 acres in Scott County. There's about 1,000 acres this year out there, again, kind of Scott to Garden area. The idea behind it is durum is normally sold at a higher base price than common wheat, although it's not traded on the board of trade. But the idea is that there's some potential value there. We were really targeting limited irrigation, and this line has performed very similar to the hard wheat Joe under limited irrigation. At this point, we're transitioning to looking more for dryland performance because we think that's going to be a critical issue going forward. But we're excited about that. I think the next 3 or 4 years here will really tell us how successful we're going to be with this if we can really get it off the ground and get it to be something commercially viable for Kansas.
There are a couple of target quality characteristics with durum that make it unique. What are those?
Yeah, so one is protein, is you need to hit the protein marker, and that's 13%, but unlike other wheat, like going higher doesn't give you any benefit, so you want to hit 13, but you don't really want to hit 14 or 15. You're just giving away protein at that point. Vitreousness, so kind of the glassy appearance of the grain, is really important, highly influenced by environment. So we had some struggles in 2024 with vitreousness because we had high humidity and a fair bit of rainfall toward the end of grain fill and during harvest last year. And then the color. In common wheat, we've tried to get rid of all the yellow color for a nice bright white flour, but in pasta, you want a nice bright yellow color. So breeding for those characteristics are important. And so I think we're in a place where we're at least okay to good in all those categories, but wanting to build to a premium quality product.
Is the durum irrigated dryland? Where are you at with that?
Right now, most of what's under production is irrigated. I think when we first started this, we kind of assumed that it would be a good target for as aquifer declined, that this would be a good target. I don't know that we anticipated how much forages and things like that would come into play. So I think in order for this to be on a scale that's sustainable, we're going to need that durum. We're going to need the dryland production. So the program now does have irrigated trials, and we still do a lot of irrigated work, but the focus has shifted to a lot more dryland work to kind of hit that niche for the dryland production, which will give us sustainability. It'll give us access to more acres.
And the reason for initiating the durum program, right now durum is grown in Arizona and California. They're having water issues, and then some in North Dakota. But why does it make sense to do this in Kansas?
Yeah, so we looked at it and we said, desert durum, even 25 years ago, it was a case of that's probably not going to be there forever, that Los Angeles and Phoenix are probably going to demand water that is now going on wheat production in the desert, and that has played out largely. And then in the north central plains, then we've also run into those issues with fusarium, and hopefully we have less fusarium in Kansas in most years. So it felt like there was a niche there. It felt like it was, and we do know that durum on average sells for about $1 a bushel more than common just on a cash price in North Dakota. So we do know there's some elevated value there. So there was some built-in value. It looked like there was some market opportunity. So those were the things that we wanted to pursue.
And there are pasta factories in Kansas City and Colorado. So
yes,
geography makes a little bit of sense there too.
It does.
So the thing in common amongst all these traits, the super high protein, the fibers, and the durum, you're just trying to add value, another level of value to maybe some things that are out of the commodity wheat pool. Is that correct?
As we've thought about it, I think it's really important that we have some things that are actually value add and that we increase the value of wheat. I think we'll always have commodity class wheat, but there's opportunities there for this value add. It will keep wheat on acres in Kansas and improve profitability. And we know having wheat on acres in Kansas is important for soil health, for the summer crops that follow it. And so all that work that Romulo has done and some of the others within the extension at K-State to show that. So we really want to make sure that wheat is a viable crop for our producers in the state. And we want to be able to capture that value. We also know that if we start pulling acres out for durum or super strong gluten or high protein or fiber or high antioxidant or whatever else is out there, then those are acres that are not in commodity class production, and you start to change the supply and demand side just on commodity class as well.
You mentioned the Wheat Genetics Resource Center earlier. I did want to touch on a couple of projects. The Wheat Genetics Resource Center has become a collaboration amongst different universities and different private companies and work on several projects, and I wanted you to comment on a couple of them. We can't forget about drought in Kansas, obviously the Central Plains, but you're doing some work, or WGRC is doing some work on something called sharp root tips, which I thought is pretty fascinating, but tell us about that.
That's actually a project that Sinish Segal at South Dakota State is leading and looking at the shape of the root tip and whether that affects the ability of those roots to penetrate more deeply into dry soil. And so we do have some materials that we know have performed better under drought. They were materials that I had started in my program back in 2015, working with Edward Akanoff. His crew did a lot of the phenotyping, a lot of the genetic work, and they've identified some genes that seem to give us a cooler canopy under drought, which means that it's somehow staying in a better water status. And it now appears that that's associated with some root characteristics. And so, I don't know if we know for sure that the sharp roots are going to be an important characteristic, but it's something that's very much definitely worth exploring and putting it into this whole package of different traits that can affect our ability to tolerate drought. And I think at that point, then we're trying to identify just the right combination of characteristics for each environment. A really, really severe drought is going to require probably a different set of characteristics than a moderate drought. But as we understand all these different traits, I think we can better define what's like, okay, this would be really good if you're expecting to be in a really, really dry situation. Or if you're expecting to be in a moderately dry situation, here's the better set of traits. But it's really just giving us the genetic tools to be able to address those in a more precise way and in a more scientific way.
Great. Well, thanks for touching on all of those, Allan. Before we wrap up today, let's touch briefly on what's coming out of your program here, maybe some recent releases and what you've got in the pipeline.
Yeah, so KS Providence picked up quite a bit of acres this year, looks like, based on the seed sales. So we're excited about it as a variety. It's done well for us. It's been very consistent. Higher yielding variety, has some scab resistance, so it's a good fit for those who are worried about scab after corn. We released KS Mako a year ago. It's been a good performer. It's a high quality wheat, so this is one that's going to be on the preferred quality lists, and it also gives us some wheat streak protection in central Kansas. It carries that WSM2 gene, so doesn't really help us much against trimv, but most of what we've had in central Kansas has been straight wheat streak, and that gives us some protection for those acres there. We have the line on increase with the high absorption. We'll have to see high Rabinos Island. We'll have to see how that gets released, whether that's a public release or an IP release. There's a set of 3 lines on increase. They're all Bob Dole by Joe sisters. They've done really well. I just had a really hard time separating them. We talked about losing some locations. I just didn't feel like I had the data set to really separate those, but I think one of those will end up getting released. They carry wheat streak resistance. They carry acid soil tolerance, really nice disease package, good yield potential, been very good yield potential, good quality. So I'm excited about the stuff that's in the pipeline.
And last question, I guess, we're recording this in late winter. From your perspective, your research crop, how does it look out there?
We're in good shape, I think, right now. I mean, I would have rather we not been as cold as we were and maybe as dry as we were out west. In general, I think we're in pretty good shape, at least relative to where we have been the last few years for sure.
Allan, thanks for joining us and bringing us up to speed on a lot of topics.
Thank you.
Thank you to Dr. Allan Fritz, the wheat breeder at K-State here in Manhattan, for joining me on this episode of the podcast. If you have any follow-up questions you'd like to ask Allan, just email us at kswheat@kswheat.com. I'm Aaron Harries. Thanks for listening.